Okay, so once again, thank you for joining us this afternoon. We have Mr. John Siefert, science and agriscience teacher at Conrad Weiser High School in Berks County. Um, his co-teacher and teaching partner in crime, Mr. Adam Sirfoss, is also joining, and I believe might be incorporated into the information shared this afternoon. This is a really exciting session that I've been anticipating. Um, for quite a while now. John has been an integral part of our steering and advisory committee for the center's year-long biotechnology and agriculture professional development program, which we much appreciate. And ever since John proposed this idea way back in, I think it was December of last year, believe it or not, I've been excited to see and for him to share what he's doing with the use of bioinformatics in his classroom. So John, the Zoom session is all yours.
All right, thanks a lot, John. I really appreciate it. I do have to. For whatever reason, my the last couple of days. So Adam, if I'm starting to glitch or whatever, if you could, I don't know, signal to me or something, I might be all right.
Well, at least on my computer, you're already glitching.
Hopefully it won't be an issue. I want to point out a couple of things here quick before. Am I already glitching? You guys are frozen. Adam, you're frozen. I can't hear what you're saying either.
How about John Seaman? Can you tell us, is it my computer? Is it John's? What's going on here?
Well, it's kind of both. You're taking turns. I think we're good now. I can see John moving. So I think we're good now. There you go.
All right. So as I was saying, we found out our internet at school is pretty horrible. So we'll do our best. I might have to turn off my screen. Maybe that'll help, just use a little bit less bandwidth. But I'll try to keep rolling here. And if you can't hear anything, either unmute yourselves and let me know. I know John said ask not to do that, but I'm totally fine if this is a pretty informal presentation. Or you could also let me know in the chat box. I should be able to see it here. Two quick things before I get rolling. I did link the presentation in the chat. So if you want to take a look at that, feel free. It should be visible for all of you to view. And if not, I can change the link setting. And then I'm going to talk about it right now, and I'm not going to show you again until the very end, but of assignments involving bioinformatics that I use in some of my classes, I also linked that folder for you. So hopefully you can get in there, and hopefully I can convince you today that bioinformatics is worth incorporating into some of your classes and how we're doing it here. So I just wanted to point out those two things. So with that, I'm going to share my screen and get rolling here. So first things first, I want to kind of declare I am no expert in bioinformatics. I was in education, and I got hired here at Conrad Weiser the next year. And when I got to Conrad Weiser, I felt woefully unprepared once I got to see the curriculum and got to see our facilities. And so most of my knowledge about bioinformatics has come from just educating myself, taking some workshops, and a little bit in my master's program at the University of Maryland. But this is really something that's just been kind of a hobby for me. And I think I found a lot of joy and a lot of success in incorporating it into my classes. So just full disclosure that I am no expert. So I'll do my best to share how we're using bioinformatics here at Conrad Weiser. So I think some of you are maybe familiar with Conrad Weiser and kind of where we're at and what we're known for as far as our ag department. We're historically a pretty rural school, although more recently the district has become very suburban. And so out of maybe 100 to 125 ag students that we have come through our department every year, maybe 2 or 3 of them have any on-farm production ag experience. So our predecessors in the department really shifted our program away from production ag and really geared us towards agriscience. And it's just what our students are going into career-wise. So we are really fortunate to have excellent lab facilities. Those pictures down at the bottom will kind of highlight that. This talk isn't really going to discuss the wet lab side, our capabilities in the lab. It's going to focus more on the bioinformatics and the computer side, which I think is great for a lot of schools that don't necessarily have the lab resources that we do. For us, bioinformatics is a really nice connection between the lab and the classroom. But I think hopefully I'll convince you that bioinformatics is not maybe easy, but it's worth trying to incorporate into your ag classes. So I guess I should start. I don't know the level of knowledge for all of you. So I'm sorry if at some points the level of the talk is too high or if I'm insulting your intelligence and it's a little bit too low. Again, my colleague Adam is here to help answer some questions in the chat box if you have any. But I'm going to start here, and then my goal is really to talk about what is bioinformatics, how is it being used, and then I'm going to spend the second half of the presentation really just kind of walking you through a few examples of how I use bioinformatics in my classroom. And my ultimate goal, like I said, is to help you answer the question, should I be teaching bioinformatics in my classes? Can I even teach bioinformatics in my classes? And then giving you a bunch of options to maybe tweak and manipulate and apply data that you think, wow, that's really cool. I think I could do something like that in my classes and probably do it better than I'm doing it. So, but in any case, like I said, we'll start here. So what is bioinformatics? And as the name suggests, it's really, bioinformatics is this emerging science. I would probably say it's an emerged science, but an emerging science that really uses computer science and mathematics to make sense of really large biological data. And so going back to the middle of the 1900s, biology was still really a field of observation or a science of observation. But in the 1980s and 1990s, with projects like the Human Genome Project, biologists began collecting enormous, what I'm mostly going to talk about today is DNA sequences. But collecting DNA sequences, RNA sequences, protein structures and sequences, and now we're going to talk about the metabolome today a little bit. So biology is now a field that collects enormous quantities of data. And there's really no way that we can make sense of that data and find patterns and meaning in it without the use of computers. So a lot of people like to say that bioinformatics is where molecular biology and computer science meet. I think that makes bioinformatics sound really intimidating to most of us. Like I know very little about coding and computer science. That's not really my forte. So my use of bioinformatics is more on the user side. How can we use the software that's available to take a look at real data with our students and try to find patterns and make sense of it? Of course, there's a huge demand and a huge need for students that actually know how to do the computer programming side, but that's not really what I'm going to be talking about today. So I want to talk a little bit more about these like, where is this data? What is this data that we're talking about? And again, this is going back to your high school biology class, so I'm sorry if this is a little bit too. The human genome is, or a genome is, the complete set of genes in an organism, and in this case, in humans. So these numbers might not be totally accurate, but to just give you a sense of how big these data sets are, or 6 billion or 3 billion base pairs. And if we look at that in any one human, it ends up being about 22,000-ish protein coding genes. So for just one human genome, that would be 6 billion nucleotides, and then trying to identify where all of those genes are. Today, the price of DNA sequencing has gone down. I think it's under $1,000 now to get a full human genome sequence. And so you can imagine every person or every animal or every plant that has its genome sequenced and gets added to this collection of data is a lot of data. So I'm going to use some words here, again, going back to your high school biology class or college biology class, but we know that in order for that genetic information to get expressed, DNA has to be transcribed into RNA, which would be here mRNA, and then that mRNA eventually gets translated into an amino acid sequence and eventually makes a functional protein. So the bioinformatics really, to me, started with making sense of the genome, but has now moved on to the RNA or the transcriptome and all of the proteins in an organism, the proteome. And so I have a question mark here for the size of the transcriptome, because there's really widely varying estimates of how big the transcriptome is. So there's at least 22,000 RNAs, right, at any given time. So the question here is, how can we make sense of all of the RNAs that any given cell in any given organism is making at any given time? So we know there's at least 22,000 for humans because there's 22,000 genes. But what we're finding is many of the genes in the human genome.
Turn off your video, please.
Is it glitchy?
You're really glitchy.
Okay. Adam just yelled at me to stop my video, so I'm going to stop my video, and hopefully that helps a little bit. So just a little bit more about this transcriptome, and then I'll move on. mRNA is the RNA that gets transcribed that's going to be made into a protein, right? So we consider this to be coding RNAs. But what we found out is that there are a lot of other kinds of RNA, a lot of non-coding RNA that is never going to actually be translated. So you guys are probably familiar with a few of these. tRNA, for example, is an example of a non-coding RNA. Its function is as RNA. It never ends up getting turned into a protein. In any case, there's lots of other kinds of these non-coding RNAs. So the real question now, even beyond the genome work that's happening, is can we identify and sequence and make sense of not the DNA, but the RNA that's in any given cell at any given time? So it's kind of a newer and exciting area of molecular biology and bioinformatics. And then finally, for those RNAs that do get translated into proteins, this number really widely varies. The old thinking was that one gene got coded into one protein. We know that that's not true anymore. So at a very low estimate, we think now that there's maybe around 92,000 human functional proteins. Chances are that's way, way higher. It's just that there's 92 that have been really identified and kind of mapped individual polypeptides that end up making those proteins. So I know I'm rambling here. I'm going to move on, but I just wanted to give you a sense of how big are these data sets and what kind of molecules are being studied in the field of bioinformatics. So the genome was just the beginning. Data sets are now being measured in petabytes. Petabytes are equivalent to 1 million gigabytes, and there are now databases that are hosting like many hundreds of petabytes worth of data. So just like mind-blowingly large data sets of DNA and RNA and proteins, and now even metabolites or the metabolome. So hopefully, hopefully that gives you a sense of what kind of molecules we're studying in bioinformatics. So how is bioinformatics being used? This is probably an abbreviated in a short list. I just want to point out a few things. The main message here is that bioinformatics has really infiltrated most other life sciences. So bioinformatics is being used in evolutionary biology to determine relationships between species, in finding new drugs, or finding drugs that work for some people but not other people. So this idea of personalized medicine. In biotechnology, obviously having DNA and RNA and protein sequences is really useful in a sense of genetic engineering. In alternative energy and waste cleanup and antibiotic resistance, more of an environmental standpoint. So using bioinformatics to find the most efficient and best organisms to produce biofuels, or maybe look at the crops that could be best utilized to produce those biofuels. And so from plant breeding, plant biotechnology, epidemiology right now is a big one that I think we're all more aware of now than we were maybe a year ago. There are tons of sequences of the various versions of SARS-CoV-2 and lots of other plant and animal and human diseases. The final one here, GWAS, or the genome-wide association studies. This is a fun topic that I discuss a lot with students in my biotechnology class. And essentially a genome-wide association study, if you're not very familiar, are studies that try to identify the genetic cause of diseases that have some kind of genetic component, but we don't know clearly one genetic cause. So an example is asthma. We know there's a genetic component to asthma, but no one clear gene that causes it, right? It's a polygenic condition. And so a genome-wide association study, as a really oversimplification, would be, let's take a full human genome of 1,000 people that have asthma, a full human genome of 1,000 people that don't have asthma. Let's align all those 2,000 different human genomes, and let's find where they're different. And not only that, let's find where they're different, and let's find the patterns of what mutations or what DNA sequences are slightly different in the people with asthma and without asthma. So genome-wide association studies have become a really common tool to find susceptibility and links with disease or conditions. So that's great, probably, but this talk is supposed to be more about bioinformatics in agriculture. And so I don't want to spend too much time on this, but suffice it to say that there is a really wide application or wide range of applications of bioinformatics in agriculture. For example, identifying candidate genes or mutations or alleles that would either give beneficial traits or a kind of weeding out harmful traits in both plant and animal agriculture. So we have a student right now, the name might sound a little bit familiar to some of you, Dylan Shoemaker, was a graduate of our program. He went to Penn State. He's now getting his PhD at the University of Wisconsin. And he is in a lab, a plant breeding lab right now, where they're breeding different lines of corn for kernel color. And then they're doing bioinformatics to determine what are the mutations that are leading to maybe more desirable colors of corn kernels. So that idea of identifying the candidate genes or mutations. Another really common example of bioinformatics, how can we look at either a group of animals or a group of plants and try to determine and maybe weed out through selective breeding predisposition for disease. And then finally, this is maybe a kind of a mind-bending one, but we're starting to use bioinformatics in the ag industry to not view corn as an individual organism or alfalfa or soy or even the animals, but instead, because we have now all this data for corn and the soil microbes and the pests that might be in and around those crops, we can look at all of those things together and start asking, well, maybe not because any of the genetics of the corn itself, but because they're interacting negatively with organisms in the soil. So this whole starting to look almost like the human microbiome with bacteria in our guts, starting to look at agriculture in the same kind of way, looking at our crops as an ecosystem as opposed to just those plants or animals. So really, the use of bioinformatics in agriculture is all about trying to produce the strongest, most pest-resistant, most nutritious crops and livestock that we can possibly produce. Okay, so I'm going to looking at some real examples. And so the question that I think I mentioned this already, that I want to try to answer for you guys is, can you and should you really teach bioinformatics in ag classes? So on the left side, looking at kind of more of the wet lab side, or on the right side, more of the bioinformatics. And I would argue both. I think it's great that we're teaching students lab skills. I love coming from an ag background and the idea of learning by doing. So I think there's a lot of value in still teaching our students the wet lab side. But realistically, moving forward, there's going to be a lot more jobs and careers for our students integrating big data and computer literacy. So I would really argue that even though it might seem more fun for our students and engaging at first, that there's real value in teaching bioinformatics in our classes. And I won't talk about this for too long. The World Economic Forum did a jobs report in 2018. There's lots of these out here. On the right side, if you look at like what is the technology and what are the kind of careers that are going to be in high demand in the United States? Big data analytics, Internet of Things, app and web enabled markets, machine learning, cloud computing. We know that we need to prepare our students more in these kind of computer literate, for these computer literate fields. So just maybe a little bit more convincing of why I think bioinformatics is really a good thing to tie into our ag classes. So the way that I use bioinformatics, I teach biology. I also teach biotechnology as part of our ag department. And then I also teach a general introduction to agriculture class. And so I don't use bioinformatics all the time in every class, but in our ag science classes, a lot of times I'll do kind of independent bi-weekly bioinformatics assignments that connect to our class topic in some way. They typically complete the assignments independently, so I'll assign them and I'll give them 2 weeks to complete it. And so I'll show you some examples of assignments like that. I really like those because it forces the students to explore and click and try and fail and redo and hopefully learn a little bit about biology and computers along the way. I also like to do just real quick demonstrations. In our biology classes, our students are getting tested on things like inheritance and genetics and point and non-point and sense and nonsense mutations. So showing in our ag classes, showing a real world connection and using those is a great thing that we can incorporate through bioinformatics. And then sometimes I'll also do larger kind of like marking period or semester long assignments where maybe, for example, students could choose a genetic condition and then they start diving into this bioinformatics software to find the specific mutations and looking at the genes and the sequences that cause those diseases. And then finally, Adam and I are both pretty passionate about helping students with independent research projects. So we try to combine kind of the wet lab side because we're lucky to have really great lab equipment. So we'll have students extract, purify, and amplify DNA through polymerase chain reaction. We'll send DNA out to labs for sequencing, and then we'll get the results back and have them do some bioinformatics analysis on it. So starting to combine the wet lab and the bioinformatics side has been really fun for us. And then I think for those of you that don't have the lab facilities, one of the best things about bioinformatics is that real meaningful data and huge amounts of it are being published constantly. So once you get a little bit comfortable with searching those databases, and once your students get comfortable searching those databases, you can conduct entire research projects with real meaningful data, and your students might be able to find patterns or meaning in that data that no one else has really thought to look for. And so without requiring any kind of wet lab facility, you can do pretty high level genetics research, which is really, really exciting. So again, I know we don't have a ton of time left. We have, I think, about 35 minutes, but I want to try to show you at least a few examples of that. And I don't know if Adam or John, I can't see faces and I can't see because I turned off my screen. I don't know what questions that have come through or if you guys are hearing me okay still.
None so far, John.
Okay, I must be doing an awesome job, or a horrible job either way. Okay, so what I'm going to do is go through a few activities, and what I'm actually going to do is kind of split my screen, and I'm going to demonstrate 4 different activities that I do with students. And if you guys either have a second screen nearby or want to try to split your screen and do it along with me, I think I'm going to move kind of fast, so it might be tricky to keep up. But if you mark them or type some things in and explore and tune me out for a little bit and then join back in, that's totally fine with me, because watching someone do this is one thing, but really just kind of playing around and using the software is the best way to learn. So like I said, I shared this PowerPoint with you, so you can go back through and do them on your own. I also have a bunch of other ones with more detailed instructions, so you can do those. Real quick though, the assignments that I'm mostly going to show you today, or the activities, utilize databases published by the NCBI, the National Center for Biotechnology Information. So depending on what classes you teach, you may have heard or may have used some of their information before, but this is a taxpayer-funded program, federal program, and so they house these huge databases. So any genetics research that uses federal research funding, their information must be posted and hosted by this database. So some of them are PubMed. This is more like access to the biomedical journals, but I'm going to show you GenBank, which is where the DNA sequences are largely posted. I'm going to show you NCBI Gene, which has slightly different information about not just the DNA sequences, but about genes in particular. I'm not going to do much with proteins today. So bioinformatics is super powerful for protein sequences, but I'm just going to kind of skip that. And then I'm going to also show you a program called BLAST, which is a tool that we can use to input a DNA sequence and then find other comparable DNA sequences. So if you, after today, if you want to spend a little bit of time just starting to get familiar with bioinformatics, I would say suite of databases and websites, but they're there and they're free. And if you can learn how to use them and share that with your students, this whole world of data and information becomes available to them. Okay, so I'm not sure if this is gonna look good or not. I'm gonna try to split my screen here. And if it's too small, just let me know and I'll try something different. But like I said, what I'm going to do is go through a few different activities. And so the first thing that I want to show you how to use is a website called NCBI Gene. And this is a real simple activity that I show my students, one of the earlier assignments that I would probably give them. And I'll just give them a gene name to search for. And then I'll ask some simple questions like, type this into NCBI Gene. What's the name of the gene? What organism does it come from? What's the location of the gene? So where in the genome is it found? And then in my upper level, exons are present, or what's the function of the gene, or what tissue or cell types express that gene. So I'm going to just walk through steps 1 through 5. If you want to do them along with me, please feel free. So I'm going to type in CSN2 and just hit search. And sometimes these databases can take a while, depending on the time of day and how busy they are. But I'm not interested in Homo sapiens. So it turns out humans do have this gene. But I'm going to scroll down to Bos taurus. So since this is an ag bioinformatics workshop, we're gonna focus more on to the search results. So these are all of the organisms that have that gene. So Bos taurus, or cattle. Mus musculus, so the house mouse, Homo sapiens, Norwegian wat, pigs, goats, right? So all of these animals have this gene. And that is a concept that I like to talk with my students about. This relatedness that, yes, we have differences in our phenotypes and our anatomy, but at a genetic level, all mammals share a lot of similarity. Yeah, if it gets really bad, let me know. Yeah, Adam stopped in and let me know that my audio is pretty bad. So I'll keep trying it on here. If it gets really bad, I might try to jump on his computer real quick and continue. So in any case, I'm going to click on this CSN identifier, and we're going to see more information than we possibly wanted to see. So some things that I frequently show, we don't have an idea yet of what this gene does, but it's a casein protein. So in this case, involved in milk. There are 8 exons, which are the coding regions of the gene. One thing that students really like to see, if we scroll down here to the genomic regions and transcripts, we can actually click on this button here, and we can actually view the A's, T's, C's, and G's, right? We can view all of the nucleotides. In this form, it's not super helpful, but we can see it, right? Which is pretty cool. FASTA format is just a format of showing a gene, and it's actually showing every single base pair or every single nucleotide in that gene as a text letter. So if I click FASTA, here we can see the entire sequence of that gene. And the beginning here tells us not only is it from Bos taurus, this actual sequence came from the Hereford breed. And again, it's on chromosome 6. So I like showing things like this to show students, again, this is one small gene in the cattle genome, but it's a lot of data, right? And so students, I think, enjoy seeing that. Let me go back here. So I think I've answered some of these questions, right? So like, what is the location of the gene? How many exons? If we continue to scroll down, there are papers published that involve that gene. I think Sarah, I think you're in here. I know your students had researched like the A1 milk versus A2 milk. So being able to find some information about those genes and helping students explore their independent projects. And then even further down, again, this gets a little bit more in depth, but what are the functions of this gene? So it has antioxidant activity, it is involved in protein binding and some other things. What component of the cell is it expressed in or expressed by? So in the Golgi lumen and in the extracellular space. So again, I don't know if that information is necessarily super helpful in all instances, but if I were doing a milk lab, for example, in our 10th grade current topics in ag class, I might take a little bit of time and come in and look at, well, what are the genes involved in milk production and what kind of species have those and how are they different from species to species?
John, I have a quick question to interject for you here.
Sure.
Is there a way to, in that database, is there a way to search for genes or proteins, sort of filtered by what cellular organelle they're found in? Like, can you do a search for all of the ones found in the Golgi apparatus, or?
I have never done that, and that's a really good question. Let's find out. Let's see what comes up. So we're getting, a wide variety of genes here now. So a lot of these are, probably have that somehow in the name, but I'm guessing there is a way to do it. I just have never physically done it. I know that you can filter by organism and by, whoops, I don't know where I'm going now. I know you can filter by species and things like that. The other thing, I didn't really talk about it, but if you don't know the name of a gene, you can just search by the function. So I don't know, let's say we're interested in, you're interested in growth hormone, but you don't know the name of the gene or the identifier of the gene. If you just search for growth hormone, you're going to get a bunch of different kinds of growth hormone genes. So I don't know if that really answers your question, but you don't have to search already knowing a gene name. You can go and explore and have your students try to find genes, kind of send them down the rabbit hole and find genes that are associated with milk production and see what happens. And I see other questions are coming through, but I don't know what I did to my screen. I can't see them. So if you guys wouldn't mind shouting those out or reading them for me.
Yeah, so Sarah Beth asked if you were able to type by search by phenotype or physical trait. But then I think you answered her question before we could even ask it. So perfect.
I will show one other, I'm going to show one other database. There's a database called OMIM, Online Mendelian Inheritance in Man. And in OMIM, that's more where you can search by a condition or a disease or a phenotype, and then it'll give you a list of the disorders. So I decided not to show this one today, but let me go and show real quick. It's all interconnected program. And I don't know, my internet must be bad because it's looking really weird, but usually it doesn't look like this. But for example, you could search by a disorder. So you could search by a certain condition, and then it'll show you, if you click on one, I'm not going to do that right now. It'll show you, yeah, it's looking really weird on my end right now. I think it might just be my internet, but it'll show you which genes are associated with those. Yeah, it's not looking right on my end, but it'll show you what genes are associated with those phenotypes. So that might answer that question just a little bit better. Any other questions while I'm still here, or before I move on to the next one?
Not seeing any.
Okay, awesome. So what I was going to show you next, let me get back to my CSN. So continue with this casein beta. I'm going to use that as kind of a continuing theme as we go throughout. So if we scroll down here, though, there's one other link that I think is really useful. So GenBank is a link that is going to take us to more probably useful information about that gene. So if you click it, it's still within this NCBI website. Here you can see exactly how many base pairs are in that gene. It's DNA, it's eukaryotic, so it's a linear gene. The species, all of its classification. And if you scroll down, there again, we see the entire DNA sequence. So sometimes I'll just give students like I wouldn't call it like a web quest, but I'll just give them a gene and ask them to find all of this information. So you'll find a few of those examples if you click into my bioinformatics assignments. And I have fun. I just pick random genes or genes that I hear of from articles, and I learn a lot about it as I'm doing it too. What I would like to show you here is kind of part 2. And so we've been looking at the Bos taurus, the cattle CSN2 gene. And over here, if you get to GenBank, there's a link to run BLAST. And so I'm going to click this and we'll see what happens. Basically BLAST, and I'm not going to click anything. I'm not going to touch anything. I'm just going to BLAST. And then while it's searching, I'll kind of explain. So basic local alignment search tool. What this is going to do is go out and search for all of the other species that have a similar gene. So again, I like to think of this as a kind of an evolutionary relationship tool, but we can also use this in research. So if there's a disease that we want to study, but we obviously can't use humans in preliminary research, we can go out and find, well, what organisms have a similar gene to humans that we can actually research. So do fruit flies have a similar gene? Do C. elegans have a similar gene? So I like to think about that in terms of model organisms as well. But so I searched the CSN2 gene. I went to BLAST, and I found this is the list that comes back. So bovine, obviously that's what we were looking at. We're looking at Bos taurus. Capra hircus, which is goats. So I'm sure since we have a bunch of ag teachers in here, a bunch of you guys knew it. Ovis aries, which is sheep. I don't know what this one is. If anyone knows, feel free to unmute yourself. Bos taurus Jersey breed, and some others. So again, a lot of the species that we would expect to see having similarly related casein genes: cows, sheep, goats, and I think pigs are in here somewhere. A couple things over here on the right, so I'm not really going to do much more here, but for any of these, we can learn some information. So percent identity is fairly straightforward. So we search for the Bos taurus gene. Percent identity tells us how similar are all of these other sequences. So this goat gene is 94.87% identical. And so I like to talk to my students about relatedness. A lot of our students don't know that you can drink goat milk or sheep's milk and then produce cheese from them. So a fun connection might be we do a lab where students try different cows, sheep, and goat milk cheeses. And then we do this bioinformatics assignment and we talk about at a genetic level or at a protein level, how similar are the proteins that make up that milk. So kind of a cool thing there. Adam can probably proclaim this. I get excited about numbers sometimes. I don't proclaim to be good at math, but I like statistics and data. This E value is an important thing for our students to know. So basically the lower the E value, the more trustworthy the search result is. So basically this column is answering the question, are we getting these search results by chance? Or are we getting them because they truly are meaningful? They are related in some way. And so 0.0 basically means that there is zero chance that we're seeing all of these other genes just due to random chance. They're unrelated. So we are statistically confident that these genes are casein beta genes. They're doing similar functions in those organisms. And so anytime we can talk a little bit about statistics and significance, I think is important. And so that's what those numbers there mean. I saw some flashing. Any questions coming through? We're looking good.
No, we're looking good, and we discovered that animal is the water buffalo.
Ah, okay. Bubbleless, boobaless. Learned something new today. Cool.
I think it would have been aqueous bubbleless. Anyhow.
I think it's boobalis. I'm not sure. It's literally crazy. We had just looked that up in animal science the other day. I was like, this relates.
That's awesome.
I love it.
Adam and I, Adam and I co-teach a lot of our classes, and we love getting in arguments about how to pronounce things all the time. So that doesn't surprise me at all that I said that wrong or differently. Okay, so that was a really, really quick look at BLAST, but I want to show you two other kind of programs or two other activities. So if you have more questions, I have a bunch of things posted about using BLAST in that Google folder that I shared. I'm going to continue talking about the CSN2 gene. And so let's imagine a scenario where we went in and we copied the cattle CSN2 gene, the goat CSN2 gene, the sheep CSN2 gene, and the pig CSN2 gene. So all we would need to do that is click on the accession tag over here. It's going to take us to the GenBank website, and then I can go click on FASTA and I can copy this entire, I don't even know what I clicked, goat, I guess. I could copy and paste all of those sequences and line them up end on end. So again, the real power of bioinformatics is comparing, and we haven't compared anything yet. So let's try it. So there are lots of different versions that do this. I like a program called DNA Subway. So I'm going to click it and open it up. It's dnasubway.cyverse.org, and it's hosted out of Cold Spring Harbor on Long Island. You can enter as a guest, or you can make a free account. It doesn't really matter, but if you want to save programs or save projects, you have to log in right now because I've created a project for us to do. And again, if you're at home or at school and want to follow along, that's not letting me log in as a guest. Let me see if I can actually log in. There we go. So going down through the list here on the left, go to DNA Subway. You can make an account, go to public projects, and you can search for projects that other people have made. And so some of these are DNA barcoding for plants, or there's all kinds of crazy stuff in here. DNA barcodes from species found at the JMU Arboretum. It's kind of fun to look around, but I'd like you to search, or I'm going to search a project that I made. Oops. That is mammal CSN 2.2. And if you type that in, you can just click the little search button over here on the far right of the screen, and it's going to bring up my project. You can make sure that it's my name, but it's there. So all I did for this is I copied the goat. It's cutting off a little bit here. Maybe I can. No, not working. I went in and I copied the cattle, the goat, the sheep, and the pig CSN2 genes. And so this program has aligned them for us. And so again, just to think how helpful bioinformatics is, if I ask you to go through and say, all right, out of these 4, which 2 sequences, the goat, the cattle, the sheep, and the pig, are most closely related? It would take you probably a lifetime to count all of the differences between all of them, and then count, tally those differences, and then say, okay, well, these 2 sequences are the most closely related. But with a program like this, like DNA Subway, we can very easily do that. So this is an assignment that I do a lot with my students. I do it oftentimes for a phylogenetic analysis, so again, relatedness between species. I've done this with plants, I've done this with animals, different crops. So once you learn how to do it once or twice, you can really just pick whatever is interesting or intriguing in your classes, and let your students run wild. So I'm going to do, so I already clicked sequence viewer, so we saw that the sequences are there. I'm just going to go over here now and do muscle. So muscle is and I'm going to expand it now so we can see the whole thing. Muscle is an algorithm that lines up the sequences. So it has lined up all 4 sequences, and it's doing what I just asked you to do. It's looking for similarities between these sequences. And it looks kind of scary and intense, but it's really not. So if you want to see, like students really like to see this, if we click the ATCG button, it's going to zoom us into the actual DNA sequences of all 4. And mine, it's kind of freezing. So if I just give it a second, it's probably loading. They're pretty long sequences. So each of these is somewhere between like 8,000 and 9,000 base pairs. And hopefully my computer isn't totally frozen. What we're going to be able to see here, so I'll explain this idea of consensus. So hopefully my computer does zoom in here in just a second, but it's going to look at all 4 of those sequences, and it's going to find consensus. So at base pair 200, if all 4 sequences have an A, it's going to say A is the consensus, right? So it makes a consensus sequence that is the most similar between all 4 of those sequences. And on my computer, it just jumped to the nucleotide view, and I see Adam shaking his head. Good. So for example, this program has artificially made the consensus sequence. And if we don't see anything, so under pig here, for example, at this base pair, I'll look at this one actually, there's a C. So C is the consensus. And because we don't see anything different listed for pig, for cattle, for goat, or for sheep, we can assume that they all have a cytosine at that specific location in this gene. If we look at the consensus for this base, the consensus is C, but the pig is different. So the cattle, the goat, and the sheep all have C. It only shows us the difference here for T. So what we could do, this has made it a little bit easier. If we wanted to now, we could go through and count all of the places where there's a difference from the consensus and start to, it would start to make it easier for us to calculate those. But up here in the corner, if we just click sequence similarity %, it does it for us. So if we look at sequence 1 here, which we know is the pig CSN2 gene that I pasted in here is 79.77% identical to cattle, 79.95% identical to goats, 79.87% identical to sheep. If we look at some of the others and sheep, their genes or the sequence of this particular gene is 94% identical and 98% identical if we compare goat and sheep. So students like seeing the actual nucleotides and they like being able to see those differences. A lot of times what I'll try to do is pick genes that are very similar so they can scroll through like this by hand and actually count the differences. and say, well, there were 5,000 nucleotides in these sequences and only 5 differences. That must mean they're pretty similar. And so the final piece that I'll show you real quick, so that's hard to visualize maybe, but there are some phylogenetic tools or algorithms that allow us to see this visually. So if we click phylip, this is one of the phylogenetic tools. And so this is going to give us, it doesn't mean for sure, but it's going to give an estimate of, based on this gene and the sequences that we've uploaded, the goat and the sheep are the 2 most closely related species. Now, remember, this is just one gene. That doesn't make that true. But if we then start looking at other and visible characteristics, we might be able to make a better case that, okay, goat and sheep are the 2 most closely related, followed by cattle and then pigs as the outgroup here. So I do this a lot in my biology class, maybe a little bit less in my ag classes, but still kind of a fun way to use a pretty, I don't know, intense bioinformatics program, but have some fun with it and find some, again, real information about real organisms that they've heard of using real data. One last thing about DNA Subway before I leave. It's supposed to be very user-friendly and intuitive. I was using this blue line, so how to determine sequence relationships, but there are other ways that you can use this. So if you input a huge sequence of nucleotides, you can actually annotate the gene using this program. You can find introns and exons. You can find the promoter sequences or predict where promoter sequences are. You can, so basically you can show students that there are patterns and trends in the genome that allow us to find genes. So there's 22,000 genes. How did we find that out? How do we know where they are? And so this would be a tool that we could use to show them that. And then if we do DNA or next and all kinds of stuff, I'm not going to show you any of those other ones today because I'm not an expert and I haven't played around with those as much. But just even if you use the blue line of okay. I think I only have one more activity to show you, and it is actually I have 2. So I know I'm running short on time. I'm happy to stay on a little bit after 4:30, and I don't expect anyone to hang out any longer. So I don't know, John, if you have a preference, would you like me to continue on even if I go a little bit over time, or do you want me to wrap it up here in the next 5 minutes or so?
You're fine to continue on, and if anybody has to hop off, no worries whatsoever. We understand. So yeah, continue on as is, John.
Okay, awesome.
And real quickly, Mr. Siefert, before you continue, for anybody that's been following along, having trouble hearing the audio because it might be glitchy, you can reach out to either me or John through email or any other form, and we'll be happy to arrange Zoom meetings at various times to address any of your concerns or questions that you might still have.
Yeah, definitely. Thank you, Adam. I've talked all about that CSN gene in animals. I want to switch gears and talk about plants a little bit. And so I'm going to take us to a database called the BOLD database. It's the Barcode of Life database system. And this is a database that is a little bit different. And it's different in that, I don't know, maybe 10 or 15 years, realized or decided that there needed to be an easier way to identify species down to the species level based on their DNA. And so they identified a few genes that we use as barcoding genes. And what that means is a gene that is really simple to sequence. It is important enough to all living things that all species have them, but not important enough that mutations disrupt them. So basically, the genes that have been picked for DNA barcoding are found in all animals or all fungi or all plants, but vary enough from species to species that we can use it almost like a barcode. So if we find a species out in the wild, we sequence the DNA or we extract the DNA, we sequence it, we can use it as a way to confirm what species we're looking at. Okay. Adam's telling me that my internet got super glitchy. So if it is really bad, I can run and hop on another computer. I don't know if anyone else, if you guys are having a really hard time, would you like me to try a different computer and hop off here quick?
John, it only seems to happen when you have something really important to tell us.
All right, I'll try not to get excited. I'll speak as monotone and boring voice as I possibly can.
You just made me laugh. I love that.
I'll keep forging on, I guess. Again, if I'm happy to do this again or talk with people one-on-one if you guys missed something really important. So in any case, let me show you how I use this database. So a few years ago, we had students interested in the labels on food products. So how accurate are the ingredients lists on food? And so what we did, and this is the kind of the inspiration for this activity, is we got bags of tea, we cut them open, we looked at the leaves in the tea underneath a microscope, and we separated ones that looked different. So nothing scientific there. We did a DNA extraction and then amplified it using PCR, and then sent the sequences out to a lab in New Jersey that is really, really affordable for DNA sequencing. And they send us back the DNA sequences in email. And so on the next slide, I have two of these sequences. Both of these were truly from tea bags. And so if you're not familiar, tea belongs to the, and let me see if I need to go and copy these sequences real quick on the right. Tea should come from the plant Camellia sinensis, which is a shrub that grows in Southeast Asia. So just to show you how the system works, I'm going to click on plant identification. I'm going to scroll down and I'm just going to copy and paste that sequence in. If you're curious, the ends mean our sequencing wasn't great. So the company that did our sequencing knew that there were nucleotides there. They just didn't know they couldn't confirm if it was an adenine, a cytosine, a thymine, or a guanine. So I'm just going to hit submit, and this takes a little while. And so remember, for tea, we're expecting, and as foreshadowing, we did not find all Camellia sinensis as the plant species that were in there. And Adam, if I kind of glitch out, you can feel free to chime in and share the results. I think your computer's working a little bit better, but I do want to wait and see if we can find a result. My computer didn't glitch. It's still working. It usually takes maybe 30 seconds, 45 seconds, maybe a minute for this to run. And as this is running, I will tell you, we've done this with all kinds of stuff. We've done that workflow, the DNA barcoding and sequencing, and then the bioinformatics piece with insects. So last year we had a student do this with spotted lanternflies that we collected here in our district, and we compared them to spotted lanternflies that had been published in Asian databases, so in Korea and in China. And some others. So here we go. So the way this typically works, it's telling us that the top hit is, and again, I'm going to probably butcher this, Spinacia oleracea, which, any guesses what that plant is?
Spinach.
Yeah, it's spinach. So this came straight from a tea bag. And the question that our students ask then is, why the heck is there spinach in tea? And so, that led us on this whole other interesting conversation of, well, maybe it's a filler because most of us don't do DNA testing on our tea bags and they can get away with it. Maybe it was an error on our sequencing and it wasn't true. So that's also a possibility. Maybe there's contamination in the shipping or the transportation. We don't really know, but that was a pretty interesting and shocking result for our students. They ended up finding a few things. We found spinach. We found wild ginger. We found a couple of grass and weed species that are native to Europe and into North America. And so, again, we can't really confirm through this test why that happened, but an interesting conversation on food purity and labeling, and again, how we can tie in our food science classes with DNA barcoding. The really famous example of this was some students from New York City did this with sushi from New York grocery stores and restaurants. And they found, I think this was like 10 or 12 years ago, that up to 50% or even more in some cases of the seafood sold in New York City was mislabeled. So a lot of vendors were selling the fish as more costly fish than what it actually was. So red snapper, it turns out, is usually not red snapper. It's some other cheaper fish. So that's kind of the example of how we can use this bold database system for doing identifications. I won't show you the other one since we're kind of over time. So let me show this last one real quick, and then if anyone has questions and wants to hang out a little longer, I'm happy to do that. The last website that I want to show you is very similar to OMIM, the Online Mendelian Inheritance in Man, but it's OMIA, Online Mendelian Inheritance in Animals. So for any of you that teach an animal science course or vet skills or something like that, I think this could be a cool place to start. It's really not intense bioinformatics. It's more just a database of genetic information. So it's a very, very easy and approachable database. You can browse by species. So 782 disorders in dogs, 549 in cattle, and so on. I'm going to click 549 and look at cattle since we've had that theme for most of the day. And it's going to bring up all 549. And I'm going to look at this gene again. I just had milk on the brain, I guess. So beta lactoglobulin. But for all of these conditions, if my computer does decide to load, it's going to show us on what chromosome is that condition or that disorder. It's going to show us not only the chromosome, but what specific mutation. Here we go. So I'm going to search. So here I can click beta lactoglobulin. The species is Bos taurus or cattle. The name of the gene responsible for that condition is PAEP on the actual identifying tag, and learn some more information. So not only is this on chromosome 11, I know that it's on the longer arm of the chromosome at the second band away from the centromere. I can talk about that in a second, the chromosome notation. And then also down here, the breed most affected by the condition is brown Swiss. Over here, the specific mutation that causes it is when a cytosine gets transversed to an adenine at the 215th base pair upstream. So again, probably more information than we need to know, but I really love taking the phenotype. Okay, so students have heard of a disease or a condition, and now showing them that research has been done to identify the exact genetic cause of that condition or that disorder. So this website is new to me. I haven't really explored it very much, but I could definitely see us doing this in our 10th grade current topics in ag class, where we do animal science units and breeds and things like that. So hopefully kind of a fun one. I real quickly mentioned this 11q28. Every chromosome, if you kind of picture, this one to me is kind of upside down, but if you ever see something like this annotation on the right, the first number means the chromosome. So this is on the cattle 11th chromosome. The centromere is the skinny part in the middle. If it says 11p, P stands for petite, so the smaller arm of the chromosome, and Q stands for something else. I don't know what the translation for this is, but Q is the longer arm. And then the numbers that follow after it are the band and the subband. So when we stain a chromosome in a karyotype, we see this banding pattern. So it's a zip code, or it's a roadmap to where this trait is on the chromosome.
John, real quick, if I could interject a question that Jane posed to the Q&A pod. Adam answered it, looks like. Never mind. Carry on.
Okay. So I'm pretty much done. So I'm happy to answer any other questions. Adam did give me a note and asked me to talk a little bit about, I don't know if someone had asked it, but the process for extracting and doing that with DNA. There's a lot of great resources. I didn't talk about it here. I didn't know if if the wet lab side versus the bioinformatics was going to be more interesting to people. So DNA extractions can be, like you can do it with material. I joke that students can do it with materials already in their houses. So if you have dish soap, meat tenderizer, and some vodka at home, you can probably do a DNA extraction. Obviously, in the lab, we use materials that are a little bit more pure reagents and things like that. I use a kit from Carolina. They have a whole DNA barcoding workflow. Let me actually see if I can go and show it here quickly. Carolina. So this using DNA barcodes to identify, and I've modified and used some different things, but it's not super cheap. And you need to have PCR to do this process. But basically, you take your plant or your insect, you mash it up in a tube, you add a lysis buffer that's going to break down the cells and denature some of the proteins, and then you separate by centrifuging or filtering. And then once you've gotten rid of all of the cellular debris and proteins, you can take that the solution that contains the DNA, and you have to amplify it. So this kit contains the primers and the other things that you would need to do kind of traditional polymerase chain reaction. And then it also gives you instructions in this manual how to send them. The company that we use is GeneWiz, and you can send your DNA samples to GeneWiz. It's like $3 a sample to have a barcode sequenced, which is crazy. So in a class, if we have 24 kids and we put them in groups of 3 or 4, for like $16, you can send out a whole class worth of samples, get them sequenced, and then have them on your computer and let them analyze it a few days later. It really blew my mind the first time we did this kind of stuff. So the only specialty piece of equipment that you need to do that would be a thermal cycler to do PCR. And I know a few, I won't name them by name, definitely have thermal cyclers that might be willing to host some people to run some samples through or whatever. We would definitely be happy to run some samples if you guys are interested in trying it. But joy to watch students go from finding something out, like sampling something out in the environment, bringing it into the lab, purifying, extracting, amplifying, and then a few days later when they see the results of their sequencing, it's really, really cool. It's very rewarding. So that's one of the ways that we do it. So that it's a pretty user-friendly protocol if you're looking to get started. So slide. Hopefully I've convinced you that bioinformatics can be pretty cool. And I think it's teaching students these future-ready skills that they're going to need to have. They can pick the problem. It's authentic to them. It's real data. They're making connections to their other classes or their life. Of course, they're learning by doing, not only in the lab, but also by on the computers and finding trends and patterns that people haven't seen before. And I really do think it can be very frustrating. It doesn't always work and they don't always know what to do, but it forces them to think critically. And I'd like to think that it's interesting enough that they care about trying and investing a little bit of time and then giving up because it's too hard. So I've posted a few websites and databases. So Bold, DNA Subway, Online Mendelian Inheritance of Man, all of the NCBI stuff here. There are hundreds of other databases and websites that you can use, but these are some of the ones that I found. Very much for attending. I really enjoyed putting this together, and I'm happy to answer any questions that you might have.
All right. Well, thank you. That was wonderful, John. And I've been having some chats with folks privately in the chat box, and you are really getting some heads spinning and firing up some ideas and giving some folks some ideas as to ways they can incorporate this into their own classrooms. For those of you that are doing the year-long biotech program, I hope you saw some building of the concepts that we covered in the biotech basics workshop revisited, and why you need to do some of those processes such as PCR, et cetera, and then how you can then use them in further applications that John just covered. Any questions for John, Adam, before we adjourn here? I'm going to stop the recording.